Hello, Dr. Rahul. Good evening and welcome to the
Hello, Shri
webinar.
Gauri. Hello, Shri Gauri. Good evening. You can hear me, right?
Yes, sir, we can hear you.
Okay,
Would you like
fine.
to test your slides if you can share? If there are,
Yes, yes, yes. Thank you, please.
I'll stop sharing.
Yeah, just hold on. Yeah, Shri Gauri, is it visible and uh, the transition is okay of slides?
Yes, sir, we can see both.
Okay, thank you. I'm
Okay,
stopping.
okay, I'll share. Okay, so one participant has introduced. So Dr. Shaim is from Iraq and is an assistant professor in molecular biology. Thank you so much, Dr. Shaim, and welcome to the webinar. For those of you who have just joined with us, we will be starting with the webinar in about a few more minutes. We are waiting for the rest of the participants to join. And meanwhile, we are waiting. It would be great if you can introduce yourself in the chat box. Please feel free to let us know where you're from, what is your background, and what are your research interests. Okay, so I can see that participants have started joining. So without further delay, I'll slowly start with the introduction. Thank you so much, Jivisha. So, hello everyone, a very warm welcome for today's webinar on sequencing laboratory. industry NGS practices, and career insight. In this webinar, we'll explore the evolving world of next generation sequencing and its growing relevance in research and industry careers. Before we begin, let me take a brief moment to introduce you to the team behind the webinar. We are a global bioinformatics and data science company with roots in India and the US. And over the years, we have had the privilege to collaborate with leading universities, research institutions, and industry partners worldwide, delivering innovative learning and research solutions. And our mission is to bridge the gap between data driven research and skill development. To achieve this, we work with regional and international experts who help us tailor our programs to the specific needs of students, educators, and researchers. With the growing demand for skilled bioinformaticians and data scientists, we have developed a range of training and research offerings. These include online courses, specialized training programs and workshops, internship and fellowships, research services, and even degree program. Now, the student learning journey begins with hands-on courses or training programs that is suitable for all levels, whether you are a beginner or an advanced learner. And for those looking to go beyond, our flagship research program provides a platform to work on real projects with expert mentorship. We offer courses and example projects on a diverse range of topics from precision medicine, oncology, infectious diseases, metabolic disorders, to even agri-bioinformatics. And these courses are enriched with engaging content, including visuals, infographics, and video lectures that simplify complex bioinformatic concepts. We also focus heavily on experiential learning through short tutorials, practical demonstrations, and coding exercises to enable learners to build technical expertise and confidence. While our courses are fully automated and lesson based, we understand the value of mentor guided instructions. That is why we also offer workshops and training programs that combine instructor led sessions with hands on walkthroughs to reinforce key concepts. And this includes a combination of expert video lectures, interactive tutorials, and step by step hands on practicals. Now, these are just a few of the many positive testimonials we have received from participants from across our workshops and training programs. And we are also thrilled to announce a brand new 6-week online course titled From Sample to Sequencer, a Practical Guide to NGS Workflows. And this online hands-on course will be led by Dr. Rahul Ramekar, who is also the guest speaker for today's webinar. He is a seasoned clinical genomics expert with over a decade of hands-on experience in NGS, single-cell research, and translational genomics. And over the 6 weeks, participants will explore key components of the sequencing pipeline, including NGS platforms and their applications, sample preparation essentials, library preparation techniques. We will also cover experimental design, targeted sequencing approaches, and real-world case studies. And to ensure that participants are able to engage and get an effective learning experience, the course includes diverse range of features such as live mentor guided sessions, supplementary reading materials, and at the end of the coursework, students will be also able to get a certificate of completion that they can later on add to their resume or on their LinkedIn profile. We'll be covering more about this course towards the end of the session, but this is just a quick notification or an announcement for those of you who have joined us for the first time. Now, with that, I'll quickly stop sharing and reshare again and take all the participants to the OmicsLogic Learn portal where they can explore each of these resources that I just covered. Okay, I hope you can see my screen. I'm sharing the link to the platform in the chat box. Now, most of you might be familiar with the platform, may have been part of our previous workshops or programs, but for those of you who have just joined with us, this is the online learning portal for OmicsLogic. And for those of you who are signing up for the first time, you can just click on the login option and click on create an account, and you can enter your name, email ID, password, or you can log in using any of your existing social media handle. Now, for those of you who already have an account, simple process, just enter your email ID, password, and you can log in. Since I'm already logged in using my account, I'll log in using my Google. Now, once you sign in, the very first interface that you see is the course library. So here you can explore our diverse range of courses that I was covering in my previous slides. So you can explore it by topics like bioinformatics, genomics, transcriptomics, machine learning, Python, R, metagenomics, cheminformatics, single cell, and so on. Or you can also type in a specific keyword that you're interested to explore, such as COVID, let's say, and it'll show up the different example projects or coursework that are covering this topic. So this is just a quick introduction that I wanted to give you. Now, without further delay, I'll pass over the stage to the speaker for today's webinar, Dr. Rahul Ramekar. Over to you, sir.
Thank you. Thank you, Shri Gauri. I will share my screen. Okay. Shri Gauri, again, just confirm you're seeing the slides.
Yes, sir.
Okay, thank you. Thank you very much. Thank you very much, Shri Gauri, for the kind introduction. Uh, good good evening, good morning, or good afternoon, wherever you are present. Welcome to this webinar. The topic what I've selected is the breaking into multiomics and why NGS matters. Okay, so this is where we will discuss few aspects of next generation sequencing, why, how, where. Okay, just allow me to introduce myself. I am Dr. Rahul Ramekar. So after completing my master's in biology, actually, I started my work as a molecular biologist. And I worked a half a decade as a molecular biologist, and then I got an opportunity to complete my PhD in quantitative genetics. So when I was doing my research in quantitative genetics, uh, one of the prime question was how can I go deep and get some insights into biology. How can I go deep means I'm talking about the AGCTs, and then how fast and with high throughput can I get those data. So this is where I was introduced to NGS, and from my postdoc, I started a mission to work in NGS to understand, to implement, and to apply NGS in my research. So I will start. What I will do, let's start with the first question: why NGS, and with help of a case study. Okay. Now imagine a newborn girl. Okay, so who is just a few days old, and but she's having a continuous seizures. So when she's having continuous seizures, she can't feed. Okay, she can't feed on her mom, so it's very we'd say I mean detrimental for her, right? When her brain scan was done, it was inconclusive. Okay, her routine blood test normal, but her tiny body, I mean, and with the seizures every few minutes, it did not help her. She was always dependent on feeding tubes, and also the doctors were mostly left guessing what was the issue. Now, imagine being the parent in that moment, right? Or let's say even also imagine you are a clinician. So you're doing everything by the book, and let's say nothing is helping. So what do you do? And when you're out of those tools, the time is of essence, right? Time is slipping because she's a newborn girl. We need to save her, right? So this is the real story of Sebastiana. Hold on, I will just have the pointer option. Yes, right. But of course, what actually helped her was something faster, something smarter, and also deeper. This is what I was talking about NGS. So a rapid whole genome sequencing helped her to overcome those. I mean, to get the results or get to know what actual was causing the seizure. With help of rapid whole genome sequencing, what they did was within just a few days, the team of the clinician knew what was wrong. There was a single letter change in her DNA. And that was rarest of rare, right? So, but that rare mutation explained everything to the doctors. So a mutation was in a specific KCNQ2 gene. And this wasn't just a data, actually, this was a diagnosis, a rare epileptic condition, what she had. And because of this, so we knew why and how, right? Her medication was switched immediately. And fortunately, the seizures also stopped. The feeding tube came out and she went home, I mean, healthy, and she's growing very well. So and of course, this is not a fiction story, this is a real story. Okay? So this is how actual NGS has an implementation, and we will see in few of the slides. Right? So with this case study, a basic question comes in our mind, that why is that the two people, let's say we had exposed to COVID, right? So why the two people who are exposed to the same COVID strain can have completely different outcomes? One, for example, let's say ends up in ICU, right? And you have seen that. Whereas the other person not even develops a cough or any kind of effects he has. Okay. Again, why do patients with the same cancer, the same type of cancer, take the same drug, yet only one of them responds while the other sees no benefit? In plants, let's say, why do some crop species withstand drought or pests, right? While others requires a lot of care. And let's say the most important question is, how do we unravel the individual building blocks of disease? Right? And of course, so these are not the let's say medical mysteries, but these are a puzzle. And if we ask, and let's say at the core of these mysteries is the simple fundamental answer: the genetic variations. You have all heard, right, that no two individuals, whether humans, plants, or microbes, are, they are genetically identical. You have heard a lot, right? So our DNA holds millions of those tiny differences. So these differences, or let's say these genetic variations, shape everything from the color of your eyes to making it to you unique from me or from others, right? To our risk of disease, and also how we respond to the medication. Some of these variations are inherited, or also they are passed down through generations, right? Others, they arise spontaneously. But together, all of this, the genetic variation holds the key to the answer what we had initially asked. Right? And so what are those genetic variations? So these are the let's say the building blocks we know, AGCTs, right? So the variation in those AGCTs between two, let's say two samples, two persons, right? So those are the genetic variations which makes us all unique. Okay. Sometimes what happens, I mean, not sometimes, of course, these variations in these nucleotides, they results in different form of genes or alleles. Now, but let's say, how exactly do these tiny changes or these genetic variations at single nucleotide level, right, changes in our DNA that translates into such a large difference, right? There is so much difference between us, right? So this brings us to what we call into the central dogma of life, right? the foundational principles that genetic information flows from DNA to RNA to protein, and the genetic variation, what we have discussed earlier, can ripple through this central dogma. So DNA, the blueprint. RNA, we are talking about the messenger, and the proteins are the final product that perform all the biological functions. Upon this, there is also epigenetics that adds another layer, of that complexity, regulating which genes are turned on or which genes are turned off. Okay. So, and understanding, let's say, understanding genetic variation at each of this level helps us to answer different questions. Okay. Let's say we are understanding the genetic variations at DNA level. So what does it say? Okay. So is the genetic variations associated with disease, right? Which microbial species comprise a different a community, right? When we sequence microbial DNA from the gut, skin, or soil, what we do, we begin to understand not just who is there, but also how they interact and influence or the health, immunity, or even say in plants, the crop productivity. So genetic variations also helps us to understand that is the chromatin open or closed at DNA level we are talking about, right? Is the promoter methylated? When we are talking about at RNA level, right? So at DNA, which is static, let's say, it tells us what could happen. At RNA level, it tells us what is happening inside a cell at a given moment. So which genes are differentially expressed between conditions? For instance, let's say, how does the gene express in cancer tissue, which differs that in the normal tissue? We can also ask the inflammatory genes, are they overactive in one group of patients, or are the immune pathways suppressed in another? There are also novel transcripts, fusions, isoforms, which we need to understand. So these RNA level, let's say the events can be missed entirely at the DNA level, but they have a profound functional implications. And to add another level of complexity are the proteins, which are much more near to the phenotype. They are the enzymes, the receptors. So understanding how genetic variations play out at protein level will help us to give a clear picture of the function and also dysfunction. What are the relative abundance of the proteins? Are certain proteins interacting with each other? Right? For example, a single variant can alter how protein folds and whether it binds or fails to bind to the usual partners, to the drugs. And this is why proteomics, especially when we are integrating with genomics, when I'm talking of DNA and RNA, right? So it gives immense power to our data. And of course, there is another level as well, the microbiome. Okay. So what we call it as metagenomics. Okay. So all of this, what we say, the DNA transcriptomics or I mean DNA, RNA, or protein, they come under one umbrella, what we call it as omics. Okay. So they have genomics, transcriptomics, proteomics, epigenomics, microbiome, let's say metagenomics. We have. Okay. So it's quite universe we have here. But by now you might be wondering, how do we actually explore all of these layers of, let's say, at biological level? How can, or which technology can give us insights into DNA mutations or RNA expression, and even the gene regulation? Or let's say, can we decode the complexities of life with unified approach? So this is where today's topic we are discussing is the next generation sequencing, or NGS technology. So at when we discuss about at the DNA level, so here NGS allows us to identify those variations, genetic variations, what we started with, the SNPs, the indels, the CNVs at certain level. Okay, it also helps us to identify those mutations that are driving those disease. Okay. There are different germline mutations, there are somatic alterations, there are microbial, I mean, we can identify microbial communities through metagenomics. At RNA level, NGS helps us to identify those gene expressions, right, at different level, at different conditions, the alternative splicing, the isoforms, the fusions, and also the let's say the transcriptomic signatures of healthy versus disease conditions. At epigenetic level, next generation sequencing helps us to identify those, the methylation profiling or the chromatin accessibility, and even histone modifications. So at each level of the central dogma, what we have discussed, NGS is a unified approach which is currently used. So yeah, this is where actually I wanted to explain. This is at different technologies. Let's say, for example, 10x genomics. So where we are talking about the single cell technology. Right? The readout method is NGS. When we are talking about the proteomics, where Olink is playing an important role for targeted based proteomics, NGS is a readout method, and at different level. So NGS is a universal readout method at each omics level. And while most of us, even I was in assumption that next generation sequencing is more skewed towards research lab. Hey, when coming to a clinical sector or working in clinical sector, I feel that NGS has more power towards diagnostic sector. So NGS is no longer just a discovery tool. It has a powerful, or let's say it's a powerful engine that is actually driving the real world solutions. For let's start with the first one, the health and diagnostic sector. So here, next generation sequencing is actually revolutionizing how we diagnose, treat, and even prevent diseases. So here, actual the idea is to have the core of those personalized medicine. Let's take an example of oncology. Oncology, or let's say take in health sector of cancer, right? So NGS can now perform a liquid biopsy. If you have heard, this is a simple blood draw, or blood is drawn from a patient. From that, we can capture the circulating tumor DNA. That allows us to detect the cancer driving mutations. Remember, it's non-invasively. Okay? And this is happening now, and it's also evolving. But not only for in healthcare and diagnostic sector, okay, where the world, with the growing population and changing climate, next generation sequencing is also critical in the agriculture and livestock sector. Yeah. A fantastic example is of the crop development. Now, with help of NGS, scientists have done sequencing full genome of countless rice varieties, and also are doing with of many varieties. With those whole genome sequencing of those the crop varieties, they are pinpointing the exact genetic traits that can allow, for example, to develop drought tolerant plant. So by identifying those quantitative genes, breeders can develop new resilient strains or varieties, yeah, much faster than the traditional methods. And not only in the agriculture, or let's say not only for humans or plants, next generation sequencing is also key to understand the life actually that is invisible to our naked eyes. So when I'm talking about our naked eyes, I'm talking about the microbes. So consider the health of our oceans, okay, in the ocean. So using this technique called the metagenomics, the researchers can take a single, let's say, a water sample and sequence all of the DNA within it, within that the different community you have. So this gives them a complete snapshot of the marine microbiome, or the bacteria, the virus and fungi, that form the base of those aquatic food web. And mind you, this is very crucial for monitoring the effects of climate change, or even detecting the pollution, or 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 even understanding the overall health of our ecosystem. And and like I've mentioned earlier, that NGS is not just a tool for academics or research settings. The powerful application is in pharma industries or biotech based industries, is to understand the drug resistance. So, for instance, in cancer therapy, tumors can evolve to resist treatment. So, with help of NGS, I will not say exactly how to pinpoint, but with help of NGS, at least we can track the clonal evolution in real time, how those tumors are evolving to resist the treatment. So by sequencing the tumor cells over the course of the treatment, right, or over the time, we can see which new mutations are emerging. And then based on that, we can understand why, or we could understand why a drug is failing. And that will help us to make the informed decision, or we say switch to more effective therapy. Of course, it can also be used for criminal investigation. Yeah. So we are all familiar with the DNA fingerprinting, but NGS here for let's say in forensic department, it takes to a new level. Yeah. Let's say, for example, you have a confiscate. I mean, some of them have some items, right? Let's say piece of ivory or leather product, right? And you can sequence from that sample and match it to specific endangered species, and even you can sometimes pinpoint at its geographic origin as well. Right? So NGS in forensic provides hard scientific evidence which is needed to, to prosecute those traffickers, and also to protect the animal endangered species or populations. So for me, actually, I would say that NGS is not a monolithic tool. So I would say it is a foundational technology that is, reshaping the entire or many sectors, many industries in current time. So and I would say that it's not just to know what can we do, right? It's actually we can do many things with NGS, and it's not just limited to this few sectors. And I will give you one example here how actually NGS have evolved. So you all know that we have done whole genome sequencing. It's a quite a huge milestone for humanity. Now, the basis of the whole genome sequencing for human was the modern Sanger sequencing method. So uh, if so, there is, I mean, if we had done a whole genome sequencing at, let's say, 30x coverage with only one instrument of modern Sanger, I mean the sequencer, it would have taken us at least 100 years to complete. But what we have done, we had completed it between around 22 years. We have also added now the telomeres section of the chromosome. Right? But how I mean, this is one comparison. But if we take the current technology, right? So this is the current short read sequencing from Illumina, the NovaSeq X series. If we are considering using this method, the entire genome can be sequenced in 4 to 5 days or 3 to 4 days at least. There will be, of course, there will be challenge of data analysis. That is another part. But comparing it with the base model, let's say how sequencing have evolved and how, and based on those high throughput data, we can get a lot of we can do a lot of sequencing in quick time, right? And the initial example, the case study what we have seen, right, of Sebastiana. So this is one the throughput, the productivity, the fast nature of sequencer, it helped her, to save the life. But of course, I would like to quote this, that with great power comes a great risk of confusion. Yeah. Especially if we don't understand the methodology behind what we are doing. So, for example, let's say within the genomics, so within the we had started with the central dogma, the DNA, right? DNA corresponding to genomics. NGS can be applied in different ways depending on what type of question you are asking. Whole genome sequencing, whole exome sequencing, or even targeted sequencing. So in whole genome sequencing, the most this is which is the most comprehensive way to study, because it covers everything, the introns, the exons, whatever we have, right? But it also generates enormous data. And on the other hand, whole exome sequencing focus on just the coding regions, about 1 to 2% of the genome. Uh, of course, it's an efficient way for diagnostic. Adding to the layer is the targeted sequencing, where we are zooming in on specific genes or panels. So the example we took of the cancer related genes, right, or drug response genes, we are focusing on those or zooming in those targeted regions. Quite cost effective method. Now the choice between these three fundamentals, right, I mean, it's very important to understand how and where to use. If you choose the wrong one, you could miss your discovery entirely, or or you can also generate a mountain of irrelevant data. So not only for genomics, the challenge, I would say, isn't it's not sequencing only one genome, it's sequencing many. In case of the microbiology, right? So we are talking about number of genomes in one sample. Let's say we want to analyze the microbiome of soil, which help us to understand its health, right? In the conditions. We can't just use a human reference genome. We have to choose a different strategy. Different strategy means maybe 16S RNA sequencing or shotgun sequencing method. So the important question is that even before we start sequencing, we already have to make a lot of strategic choices. So strategic choices based on how to design the experiment, but also the workflow. So once you have chosen your approach, once you know which method, maybe if I want to focus on DNA, RNA, or epigenome, right? So, or in DNA, if I want to go for whole exome sequencing, right? Once you have decided your approach, now your next task is to execute that workflow. And the each step in this workflow is actually let's say if you have lack of expertise, right, it can derail the entire experiment. Just to give you information, when we're talking about NGS workflow, irrespective of whether you're going for short read sequencing or whether you're going for long read sequencing, the methodology somehow remains same. Somehow means, of course, there will be little bit changes, but the crux or the basics are same. So when let's say for the step one, right? So step one is sample preparation. Sample preparation, it can be different type of samples. You can take a soil sample, you can take FFP tissues, you can take blood samples. From there, you can isolate DNA, you can isolate RNA, right? But this is your initial step. This is where the experiment starts. And for NGS, a very famous saying is garbage in and garbage out. So whatever quality samples or quality nucleic acids you are taking in will give you a very good, better results. So if your initial DNA or RNA is of poor quality, low quality, or contaminated, no amount of your fancy sequencing technology can save your data. So remember, the step one is deceptively simple, but is the often, let's say, the single biggest point of failure. Then it comes the second step, the library preparation. Yeah. You have to prepare your DNA. I will only take one nucleic acids for sake of discussion. So if you have to prepare the DNA for sequencing, you have to make it compatible with the sequencer. And to make it compatible with the sequencer, we prepare a step called as NGS library or library preparation step. Okay. So this is quite a multi-step molecular biology protocol where what you do is you chop your DNA, or let's say you fragment your DNA and then attach special adapters which you also amplify, right? And then you make it compatible with the sequencing. And I would say, working with let's say past decade, I've seen there are different type of library prep kits, methods, and each of them needs optimization for different sample types, or let's say even for applications. And if you do any let's say any small mistake or wrong choice here, you can introduce a lot of biasness that will corrupt your data. So when we are talking about the step one, the extraction, the garbage in, garbage out, this is where right approach for library preparation will help you to avoid the corruption of your final data. And then step three, the main event, the sequencing itself, right? So whatever libraries you have prepared, it is loaded onto the sequencer, right? And this is where you would get your millions and billions of your DNA fragments sequenced. So I would say sequencing, of course, the technology to understand, because there are the evolution of the technology has happened in sequencing field, right? So let's say from the first generation to the next generation sequencing and to the latest one, right? The technology has evolved, but I would say for better experimental design, most probably you need to focus on the step one and step two. This is quite comparatively, it's more a robotic way to go forward for sequencing. Yeah. And then, of course, the final is your data analysis. Most of you most of you in your career are more inclined towards bioinformatics, of course, and I would also say that it's quite challenging, because this is where you get those millions of sequences, the A, T, G, C's, right? Those short sequence or long sequence, which you need to align them to the reference genome if you have, or if you don't have, then you have to generate those reference genome in novel way, right? You have to identify those variants, right? You need to interpret them. You need to find those actionable mutations, which in the case we saw in the first case study, where in that particular gene they identified the rarest mutation, which help her to change her medications. So for NGS, I mean, you can see like uh, the method or the concept of NGS is quite, interesting, intriguing. But the journey from a biological sample to, let's say, actionable insights is not linear at all. So it is layered, technical, and often let's say overwhelming as well. So here the real chaos in NGS is not sequencing itself. It's the dozens of decisions that surround it. So which method to choose, how to prep your samples, how to design your experiment, and more importantly, let's say, how to make sure you're interpreting it all correctly. But nevertheless, as we have seen here, for NGS, yeah, it is being applied to lot of different sectors, and it is actually powering the innovation, and I would say it has gone beyond the lab now. Of course, it has its research aspect, as well as applications, as well as clinical applications. Okay. And I would just say that in case if you want to learn NGS, it's not just about clicking the buttons or just following those protocols, right? So it is about understanding the why, the how, and let's say what if at each step. Step means, let's say, the methodology. Which methodology should I use, right? Or which library prep method, or the protocol, how to optimize it, what are the nitty-gritties of it. These are the important aspects you need to learn, you need to understand, so that you can apply it in your research, or in your current position in industries if you're working in. So this is where actually what we have done, because there are a lot of like I said, decisions to make. And this is why precisely what we have done is with help of OmicsLogic, we have designed this course. Right? We designed this program. It will help you to navigate the complexities what we have discussed, right? And it will help you to take decisions or make the technology more accessible for your work. Okay. So what we have done, we have developed a 6-week program. Yeah. And in week 1, what I'll be teaching is introducing the NGS and latest technologies, where we'll start with the big picture. Cutting through those jargons what we have to understand how different sequencing platform works and what actual future holds. In the second week, the NGS workflow, the sample preparations, right? So we have talked about that garbage in, garbage out concept. In third week, we'll be diving into the library preparation techniques for DNA, for RNA, right? Slowly, once the foundation is built, from there we will be looking into the advanced library prep method. So there are many methods available, and but to understand those concepts which will be useful for you. And of course, don't forget about the quality control. It is very important. And then in the last 2 section is where we will be working on the experimental design, and we'll be focusing on sequencing. Right? Targeted sequencing specifically. Why? Because I personally feel that NGS has a lot of potential in the healthcare sector. Right? So a lot of targeted panels are available, how they are being used. So we'll have a uh, real world case studies. But the thing what I want to discuss with you is, even though, of course, the main objective for this program is to give you a solid practical foundation in NGS. But to be honest, my primary goal, because working in this field for past many years, I would say, right, the my primary goal is something different. So my role here in this program is not just about let's say, not to teach you just of the protocols, or walk you through the workflow. My main primary role is to instigate that curiosity that will help you to ask better questions about biology, or let's say better questions about the technology, and what the possibilities lies in between. I truly feel now that in the spectrum, NGS is not at the end. Actually, it is the gateway to multiomics. Whether you're talking about DNA, whether you're talking about RNA, and in some cases protein, or of course that epigenetic landscape, understanding how to navigate and integrate these layers will definitely define the future of the science and diagnostics. And my goal here to design this program is simple: to help you just not to learn this NGS, but actually to leverage it for innovation, to create awareness. And even though I've seen that mostly NGS is being used in research, but to research in smarter way. Yeah. So with this, I would like to conclude the webinar, and thank you so much for your time today. I hope this session have somehow sparked new insights and curiosity in you, and hopefully you're leaving with something valuable to think about it. Yeah. Thank you very much for your time. Shri Gauri.
Yes, sir. Thank you so much, sir, for the insightful presentation. And I hope participants might be having questions. So please take this moment to start sharing your queries in the chat box. And meanwhile, I'll share my screen. I can see that some of the participants had joined a little bit later towards the session. And Dr. Rahul gave a quick overview about the 6-week online course, the topics covered, and just to build upon some of these concepts that were covered. So we have a detailed brochure. I hope most of you have this brochure. In case you do not have it, we'll be sharing it over an email at the end of the webinar. So please keep an eye on it. And this brochure contains details about the schedule, the topics covered, and also the various resources you get access to. So the course will be led by Dr. Rahul, like mentioned, and it will be conducted over a period of 6 weeks. And we want to ensure that all the participants are able to get the maximum out of it. So all the lectures will be also recorded. So in case you're not able to join the live sessions, you will be making the lectures available on the platform so that it can be later reviewed. And also the lectures will be available for a period of 60 days. So even after the sessions are over, you still get sufficient time to go over it. And after the sessions are over, if you have any questions, you can also email us. We will be having the lectures interactive so that when you join the session and if you have any questions, you can directly ask. But let's say you have some queries even after the sessions, and we'll get back to you. So this is the detailed brochure. Like I mentioned, we'll email it over to you, and it has the complete schedule, the dates, the time, and the topics covered. And I'll take you to the program page where the enrollment can be done. I'm sharing the link in the chat box. So if you head over to this link, it'll redirect you to the course enrollment page, and you can scroll down below and you will see the interactive session syllabus. So this is just a quick summary, but for more comprehensive topics, you may refer to the brochure. And as you scroll down below, you will see the option to enroll. So here you can change the currency based on your location, that is India or in INR or in USD, and just click on Buy Now. And this will redirect you to the checkout page. And as an webinar attendee, you will be getting an exclusive discount that you can apply. The code is NGS25. We'll also share it in the chat box. You may apply this code and you'll be getting 25% off. And you can just enter the user details, such as your phone number, country, occupation, and just select your payment mode, PayPal or Razorpay. Now, suppose your preferred payment mode is not available on the portal, you may connect with our team. We'll share the contact details in the chat box, and our team can guide you with the alternate options that you can pay for. So that's all that we wanted to cover. Now, if you have any questions that you'd like to take up to the mentor, we'll be happy to take it up. You may also unmute yourself and ask, or you may post it. Both are fine. There were a few queries about the webinar recording, so just keep an eye on it. We'll be sending it out in about 2 to 3 hours. So, yes. I did not get the query from Fadwa. Could you please elaborate? For all as in?
Shivari, I guess Fadwa is asking about recordings of the session.
Okay, okay. Yes, for those of you who have registered for today's webinar, everyone will be getting the recording link.
So even like in case if you don't have any questions now, you can also later you can ask. But the thing is that you have to think how NGS can be applied wherever you are working. For example, let's say when I was working in like I said, for quantitative genetics, it took lot of time for me to gather those data. Okay. And this is where I got to know about NGS, and I tried to integrate in my research work, and I was introduced to this wonderful technology which has a lot of good applications. Okay. But anyways, uh, I mean, you will have a lot of questions, but please feel free to ask.
Yes, sir. I guess that will be all, sir. So thank you so much, sir, for taking the time out of your hectic schedule and sharing the insightful presentation. And thank you so much to all the participants for joining today. And yes, thank you so much, Prima. And we look forward to seeing you all in the coursework. Bye. Have a great day.
Thank you all. Have a good night. See you. I'm signing off, Shigori.
Yes, sir. Good night.
Good.